Hi students, welcome to the third topic in IB biology, which is genetics. And for the first part, we will together study about genes and chromosomes. So let's start with the review of what you have learned in grade 10. Do you still remember? So once upon a time, long time ago, when you were in my class, you have learned about this in grade 10. So if you still remember, the first topic in grade 10 was about the cell. And then after that, we dig deeper into what is inside the nucleus in the topic of genetics. Now, if you still remember, in grade 10, we have learned that inside the nucleus, we have genetic information. The genetic information is found in a structure called chromosomes. And these chromosomes are actually made of DNA. Now you know what is DNA from chapter 2. DNA is a polymer that is made of monomer called nucleotides. So what is the definition of chromosomes? Chromosome is a thread-like structure of nucleic acid found in nucleus of most living cells, carry genetic information in form of genes. So parts of the DNA that codes for a characteristic, it's called the gene. So chromosome basically is a structure found in the nucleus that carry DNA, and in the DNA there are genes as the genetic information. Now, what is the next one? This is called genome. So genome is the whole genetic information of an organism. The total amount of DNA in a cell, in all chromosomes in a cell. So as you have learned in chapter 2, DNA consists of nucleotides, the ATGC. So if you read all the ATGC of all chromosomes in a cell of a species, then you get the genome. If you still remember, if I'm not mistaken, I show you a picture, a photograph of a cabinet full of books, thick books. And those thick books actually just consist of ATGC, and each book is actually for the chromosomes of human. So there will be a book number 1 until the sex chromosome number 23. So that's genome. The next one that you have learned is sister chromatids. If you still remember in grade 10, we also studied about mitosis and meiosis. By the way, in chapter 1, we also have learned about mitosis, right? Now, do you remember in interphase, all the chromosome has to be duplicated? Now you know the process is called, yes, DNA replication. And then you have learned also about DNA replication in details in chapter 2. Now, this duplicated chromosome is called sister chromatids. And sister chromatids are combined or bind together through a centromere. So the definition of sister chromatids, 2 identical chromosome strands as a result of DNA replication in S phase of interphase joined by a centromere. And the next one that we have learned also is called homologous chromosomes. Now, if you see the picture of this human chromosomes, you can see that all chromosomes are in pairs. Well, I hope you still remember from grade 10, this condition or state is called diploid. So what are the homologous chromosomes? Homologous chromosomes are pairs of chromosomes that have the same shape and size and have the same genes, but not necessarily the same alleles. What are these? I hope you still remember. Anyway, we will talk about the allele further in the next slide. But what you have to remember that all chromosomes are in pairs, and the pairs have the same size and shape. But of course, except for male, the sex chromosome consists of 1 long or big chromosome and 1 short, and this is called the chromosome X and Y, while female have both chromosomes as long chromosomes. So now let's talk about gene and allele. So you have learned in chapter 2 that parts of the DNA that can be transcribed into mRNA, and this mRNA can be translated into protein. Now, this part of the DNA is called gene because they are meaningful. They can create protein. They can be read by the cell. So it means that gene is a heritable factor that consists of a length of DNA and influences a specific characteristic. So for example, if you have gene for hair, it means that this part can be transcribed into mRNA and the protein will become your hair. It could be curly hair or straight hair. Now, what is curly hair and straight hair? That is the variation of your gene for hair. Alright, it could be straight or it could be curly, and that's what we call allele. So, allele is variation of a gene that differs from other allele of the same gene by a few base sequence. So for example, to help you to understand the meaning of this definition, let me show you this picture. So this is red blood cell, healthy red blood cell, while this one is sickle cell. So the shape of the cell is not normal because of an abnormal hemoglobin. Now if you see the gene of the normal or healthy red blood cell, the base sequence looks like this. Okay, in this point you have the GAG, all right, and CTC. However, for the cell that has been mutated and become unhealthy hemoglobin, in this part it becomes CAC and GTG. So you can see that actually it comes from the same gene, which is code for hemoglobin, but it has two different variations. The gene for hemoglobin has two variations or two alleles, which is normal and then the sickle one. And the difference between these two alleles, is just a very simple one, base. Alright, so allele is variation of a gene. Again, the gene here in this case is the gene that controls hemoglobin production, and the variations are whether the hemoglobin that is produced is normal or sickle. So I'm sure now you have 6 different terminologies in your notebook. Let's dig deeper one by one of those terminology. So let's start with chromosomes. So as you have learned the terminologies, so chromosomes are made of DNA. However, in this chapter, you will dig deeper by looking at the difference between prokaryotes and eukaryotes. So there are differences between prokaryotes and eukaryotes chromosome. Wait a minute, do you still remember prokaryotes? The bacteria, yeah, it is in chapter 1, while eukaryotes are simply multicellular like us, a human, animal, plants. Alright, now I hope you still remember that. Now, in prokaryotic chromosomes, they are circular. Do you still remember in chapter 1? Yeah, we have the nucleoid. This is the part of the chromosomes, and also a plasmid. Okay, if you don't remember, just open your Andrew Allen and see prokaryote structure in chapter 1. Now, while eukaryotic chromosome, like in our cell, they are linear. So you can see here the chromosomes are linear, a line, not a circle like this. And also there are some other, there is also another difference, which is prokaryotic chromosome just consists of DNA. As you can see from here, they are just double helix of DNA. However, oh, by the way, it is called naked DNA, so it is not associated with protein, unprotected. It's just DNA, bare DNA. However, for eukaryotic chromosomes, you can see here, they are associated with a protein. The protein is called protein histone. Okay, why they have to be associated with this protein, we will see in the next slide. The reason why it must have the protein to be coiled with the DNA is because the protein histone function is to regulate expression of genes. Now, if you see, chromosome is made of a long strand of DNA, and only some parts of the DNA that has genes. So it means that in eukaryotic, since it is multicellular, the organisms are specialized. So each cell cannot express all the genes. They have to select which gene needs to be expressed based on its function. So that's why this protein has a function to open or close the gene. What I'm saying is, if the DNA is tightly wrapped around protein, it means that this gene is not accessible for RNA polymerase to make mRNA, which can be later be used to create protein. So if it is tightly wrapped around the protein, the gene, you can call it deactivate. However, when a cell needs certain gene, so the gene will be released from the protein, all right, so that the gene is accessible for RNA polymerase to make mRNA. Then why prokaryotes doesn't have to have that kind of regulation, which gene to be expressed or not? Because they are simply a unicellular organism. No specialization needed. All genes are needed all the time. No need to select which one to be expressed and not. I hope you understand this now. And there is also a fun fact about the number of chromosomes. Each species has its unique number of chromosomes. So you can see in this table, we have 23 pairs, which means 46. Now, and chimpanzee 48, dogs 78, 24 in paddy or rice, and then in a worm and a horse, there are only 4 chromosomes. All right. So what can you conclude from here? That the number of chromosomes has nothing to do with the complexity of species. Simply, each species has unique number of chromosomes. So if a cell has 46 chromosomes, you can be sure it is a cell of a human. Or in a crime scene, if you get a cell and the cell has 78 chromosomes, it means that it belongs to a dog. And you can see from here, this is called karyogram. Karyogram is a photograph of chromosome in the nucleus of an organism. And if you can count quickly, how many? Yeah, there are 46 or 23 pairs. So you can say that this karyogram is or belongs to a cell of a human. The next terminology is genome. So genome, as you can see in your terminology, means the calculation of how many DNA base pairs or the total DNA belongs to a cell. Now, of course, you are not going to count 1, 2, 3 because it's going to end up with so many digits. So that's why the unit to measure how many base pairs, so the A, T, G, C, of all chromosomes in a cell of a species, the unit is million base pairs because it's so long and so many. And again, like the number of chromosomes, well, it simply shows who you are. It has nothing to do with the complexity. As you can see that we have about 3,000 millions of base pairs, while Paris japonica, a type of the plants like this, it has 150,000 millions base pairs, while E. coli have only 5, and then the Drosophila, the fruit fly, 140. So again, each species has its unique genome size, and larger size is not necessarily an indication that it has higher complexity. So the next terminology is sister chromatids. And I believe we have learned about this in grade 10, but that's okay, let's repeat. Now, in interphase, if you still remember, all chromosome has to be duplicated, and they become an X-like structure that consists of sister chromatids. So now, they are not called as 2 chromosomes, but they are called as 2 sister chromatids of a chromosome. So remember, identical sister chromatids are counted as 1 chromosome. You are not counting this as 4 chromosomes. This is 2 chromosomes, and each chromosome consists of 2 sister chromatids. Now, however, after anaphase, when the sister chromatids are separated, so if you still remember, like anaphase here, yeah, okay, in a cell. Now, each of them is now called individual chromosome. Now they become 4 chromosomes. I know it's confusing, but, well, that's the agreement. Hang in there, we're almost done. Okay, the 4th terminology is homologous chromosome. You have seen in the previous slide that this is karyogram of a human cell. You can see that all chromosomes are in pairs based on its shape and size. All right, so all body cells except gametes, which is egg and sperm. So all body cells, including the skin cell, the liver cells, name it. Okay, all cells but egg and sperms. have all their chromosomes in pairs, and they are called homologous chromosomes. Alright, so homologous chromosome, you can see they have the same size and shape. But other than that, they also have the same genes, right? But not necessarily the same alleles, because as you may have learned in the previous slide, that gene can have variation. In year 10 you have learned it could be the dominant allele and the other one have the recessive allele. Okay, so oh, by the way, this is the picture of somatic or body cell. All right, they have all chromosome in pairs, while this is gamete cells. And a little bit more about gene, just one point, all right? So gene occupy a specific position in the chromosome. So this is the next terminology that you need to know, called locus. So what is locus? Locus is basically a specific position of a gene in a chromosome. So later on, if you go to university for majoring in genetics, you will learn later on that the position of a gene have a certain code. So for example, this is chromosome number 1, the long hand, the position is 125, and so on and so forth. So they have certain code for each of the gene to show the precise location of that gene in a chromosome, or this is what we call locus. All right, the last one, all right? This is the last terminology, allele. So what is allele? Again, as you may learn again and again, it is the variations of a gene. Now, allele differs from one another, okay, of this other genus, other allele in a gene is by one or a few bases. So it doesn't have to take, half of the bases of a gene differ in one allele compared to the other allele of a gene. No, it's simply just one or a few bases. Like in the previous slide, you have seen that the difference between normal hemoglobin and sickle hemoglobin, the recessive one, is just one base. And it is caused by mutation. So new alleles can be formed by mutation. So this is not part of your syllabus. I just give this for your information. There are many types of mutation. It could be substitution. So for example, from C become T, or insertion. So there is a new three bases inserted in this gene, or even deleted, all right, or deletion. So what caused the mutation? I hope you still remember chapter 1. Mutation can be caused by smoking, because why? Smoking has carcinogenic substance, all right? And also later in this chapter you will also learn about radiation in the incident of Hiroshima and Nagasaki, right, in 1945, which also caused cancer. All right, so allele differ from another allele of a gene by only one or a few bases. All right. So what will happen in the next video? Like any other chapter, you have learned about the Meselson and Saul, Watson and Crick, and so on. In the next video, you will see some historical aspect in genetics that help the development of this particular field in biology. All right, thank you so much for your time, and thank you so much for your best effort to take notes. See you.